Hey everyone, welcome to BioBusters. In this video, we'll cover everything you need to know for Subtopic 1.3 membrane structure of Topic 1, which is cell biology of the IB Biology HL curriculum. I'll begin by introducing the concept of amphipathic molecules. We'll also talk about Gorder and Grendel, the Davson and Danieli model, as well as its problems, the evidence that led to the fluid mosaic model, components of the plasma membrane and its structure and properties, and finally, what are exocytosis and endocytosis. Let's get started. First, you should know what an amphipathic molecule is. It's a molecule that has both a hydrophilic and a hydrophobic part. A few examples are phospholipids, integral proteins, and cholesterol. Now you know the plasma membrane found in all cells. Well, how did we discover its structure and properties? Well, it all started with Gorder and Grendel. In 1925, they suggested that the ratio of total lipid surface area to total cell membrane surface area in the plasma membrane is 2:1. Hence, they concluded that cells are covered in a layer of fatty molecules that is 2 molecules thick. This was a key piece of evidence for Davson and Danieli. The Davson-Danieli membrane model was the first model that attempted to describe the position of proteins within the bilayer, created by Hugh Davson and James Danieli in 1935. They suggested that the cell membrane was a lipid bilayer with 2 layers of polar lipid molecules arranged with their hydrophilic heads outward and 2 layers of protein surrounding the central phospholipid bilayer, basically creating a lipoprotein sandwich. However, there were later found to be some problems with it. It assumed that 2 protein layers existed because of the dark segments observed in the electron microscope, which normally indicate the presence of protein. Secondly, more proteins are largely nonpolar, so it would be improbable that they touch aqueous environment on either side of the membrane. Third, it assumed that all membranes are of uniform thickness with a constant lipid-protein ratio, which is not true. It also assumed that all membranes have symmetrical internal and external surfaces, which is not true. Furthermore, it assumed that all membranes are identical, which didn't explain how different membranes do different functions. Additionally, this model didn't account for the permeability of certain substances. And lastly, the temperature at which membranes solidify didn't correlate with those said in the model. After gathering all of these problems, scientists concluded that a new model was needed, especially because more evidence was found that showed the Davson-Danieli model was wrong. First, it was found that membrane proteins are insoluble in water, have hydrophobic substances, and vary in size. Second, using MRI and fluorescent antibody tagging, it was found that proteins are mobile, so the formation of a static layer would be impossible. Lastly, using Friis fracturing, it was found that transmembrane proteins exist. So, in 1972, Seymour Singer and Garth Nicholson addressed the problems of the Davson-Danieli model and created the fluid mosaic model. The phospholipids in the bilayer are composed of phosphate heads attached to 2 fatty acids. The phosphate heads are made of 1 glycerol and 1 phosphate molecule that are hydrophilic, so they face outwards. However, the 2 fatty acid tails are hydrophobic, so they face inwards. The bilayer is also embedded with proteins that serve a variety of roles. The proteins may be integral, meaning they are transmembrane, permanently attached, and amphipathic, or peripheral, meaning they remain on one side of the layer only, are temporarily attached by a noncovalent interaction, and are polar. Membrane proteins can have several roles. Let's go over them. They can be channels. This means they allow the passive transport of substances between the inside and outside of the cell. They can also be carriers. This means they bind to substances on one side of the membrane and change shape to transport them to the other side. If energy is used to change the shape, they're called protein pumps. They can also be used for recognition by helping the cell differentiate between self and non-self cells, which is important in triggering an immune response. They can be receptors. These usually span the whole membrane to relay information from inside or outside of the cell. And lastly, they can be enzymes, which enhance the rate of reactions that happen at the membrane level. Finally, the bilayer, in animal cell membranes only, has cholesterol. Because cholesterol is amphipathic, it is capable of inserting itself into the cell membranes by interacting with the phospholipids. There are 5 major functions of cholesterol. It maintains structure and stability, reduces fluidity, reduces permeability of the membrane to some solutes, it prevents crystallization of the membrane, and it secures peripheral proteins. Next, we have glycolipids. These molecules are made of a phospholipid with an attached carbohydrate. They maintain the structure of the cell membrane and help cells differentiate between self and non-self cells. Glycoproteins are made of a protein with an attached carbohydrate chain. They have 2 functions. Cell-cell recognition, meaning they enable proteins to recognize another cell as familiar or foreign, and cell adhesion, so they help cells attach to and bind to other cells. All of these components of the bilayer help it get its properties. Let's go over them. The bilayer is held together by weak hydrophobic interactions between tails, and individual phospholipids can move between the bilayer. Both of these give the membrane fluidity and flexibility. Also, the hydrophilic and hydrophobic layers restrict the passage of many substances. And lastly, the fluidity allows for spontaneous breaking and reforming of the membranes, also known as endocytosis and exocytosis. Endocytosis is when substances go from the outside to the inside of the cell, and exocytosis is when substances go from the inside of the cell to the outside. And that's it for Subtopic 1.3 of the IB Biology curriculum. Thanks for watching!